If IP laws and copyright laws in 1915 were anything like they are in today's day and age, then a lot of scientists at that time would wake up with a call from their lawyers. We can say that authorship during that time was fluid. That means the person who did the experiment might not get the credit for it. However, someone senior in the same lab who just heard of the experiment would get the credit. This is what led to one of the most important discoveries in the 20th century being stolen away from the person who deserved the credit for it. She was not a physician or not a professor, but a mere 23-year-old black woman chemist. Her name was Alice Ball, who discovered one of the most groundbreaking medical discoveries in the early 20th century, which was namely the first effective. Hello everyone and greetings to the audience. My name is Aditi Gaur and I'm currently a third-year student at Sri Venkateshwara College, Delhi University, pursuing BSc Honours Biological Sciences. In this video, I will be talking about an important chapter from the history of science, which is the development of the first effective treatment of a disease called the Hansen's disease, or more commonly known as leprosy. We will also understand the science behind it and how that science has shaped science as we know it today, very specifically the medical science. This is also an ode to all the women scientists whose names have been erased and removed from the history of science. Let's start with the video. Leprosy has been recorded over the past 3,000 years, across cultures and across continents. However, the turning point for this disease came in 1873 when a very famous Norwegian physician, G.A. Hansen, discovered the causative agent for this disease. The causative agent for leprosy was a bacteria called Mycobacterium leprae. The disease got its name, Hansen's disease, after the famous scientist who discovered the bacteria, which was G.A. Hansen. This was the first bacteria ever identified as causing a human disease. But the scientific issue here was that this bacteria, which is Mycobacterium leprae, replicated extremely slowly, which means it replicated once in 14 days, which meant a gradual progression of the disease and highly detrimental effect on the immune and neural system in the human body. This led to scary symptoms like loss of sensation, skin lesions, and nerve degeneration and muscle weakness. What we must understand right now is we have to view every symptom, every disease, every social cue as if we were in the early 20th century. One of the most feared diseases of that time. It was not only because of the biological implication and the biochemical aspect of it, but also because of the social cues, also because of how the society reacted to this disease at large. Because this disease was extremely contagious, or as people thought it was extremely contagious, everyone implemented three basic laws, which was isolation laws, mandatory separation, and permanent relocation. Now, this step was taken at a drastic measure, which means, especially, let's say an example of Hawaii. People diagnosed with Hansen's disease or leprosy, they were forcibly transported to this area called Kaluapa, which was a remote island type of an area which was very far away from normal civilization, which meant these people who were patients of this disease was left for dead because there was no cure at that point in time. To understand how the first effective treatment for leprosy was developed, we need to understand that how Mycobacterium leprae, the causative agent of leprosy, affects our body. What is the biochemical pathway it takes and what is the biological implications that it has on our whole body, which causes manifestation of this disease in a patient. So M. leprae does not spread through a bloodstream like any other normal bacteria. What it does is that it affects or it targets the Schwann cells. These cells are responsible for insulating the peripheral nerves in the peripheral nervous system. Once this bacterium attacks or targets these Schwann cells, what it then does is A. It hijacks the cellular metabolism. Secondly, it reprograms the immune response to avoid detection of itself. What this means is the Mycobacterium leprae, which is extremely smart, it reprograms the whole immune response, stepwise manual, to destroy and damage the human body from within. So the disease appears to be eating away the skin. However, biologically, the damage comes from nerve failure. This is why early detection and consistent treatment of this disease was essential and still is. Well before antibiotics existed, Asian places like India, Burma and China were famous for its herbal medicines, for traditional medical systems. And one of this herbal medicine was the oil from Chol Mogra tree that was used to treat skin disease, inflammation, and yes, leprosy. So when the people from West discovered or confronted this disease in the late 1800s and early 1900s, what they did was then borrow and test the Chol Mogra oil from Asian countries. Now, was Chol Mogra oil tested somewhere? Basically, Chol Mogra oil was observed to have clinical improvement across centuries of practice. Biologically and chemically, it's because that oil contains fatty acids which interfere with the metabolism of M. leprae. Specifically, it has two major acids, which is hydronocarpic acid and cholmogric acid. Both are cyclopentyl fatty acids which cause disruption in the cell wall of Mycobacterium leprae. So scientifically, A, the active compounds were correct, B, the therapeutic mechanism was valid, and C, the problem then was not what to use, but how to use this oil. Welcome to formulation pharmacology, decades before the field had a name. So now, the issue that then arises is, we know that leprosy can be treated by this oil from Chol Mogra tree. Then why did we not give credit to the person who said, okay, let's start using Chol Mogra oil? The issue was we did not know how to use this Chol Mogra oil because the human body is a water-based system. Chol Mogra oil is hydrophobic. It refuses to mix. So when it was used raw, it can be, let's say, A, swallowed. What it then did, the observations were it led to vomiting and nausea. And second, if it was applied to skin, it did not penetrate the skin. Third, if it was injected as an oil, it remained clumped at the infection site, which caused swelling, which caused pain and inflammation. So the medicine worked, but the treatment failed, but the delivery failed. That is the turning point of history of science. The realization that a cure can exist, but still be unusable. From here, the scientific challenge, the real scientific challenge we will be discussing today was birthed. How do we change the physical being of the drug without changing the chemical identity of its active molecules? That's not pharmacology, that's biochemistry. And that is where my friends Alice Ball, our groundbreaking black 23-year-old women scientist, enters.
We have already discussed at length about the issues scientists faced with Chol Mogra oil during that time. Now we will discuss about the Ball method, the chemistry of how the Ball method actually worked, the chemistry behind how the Chol Mogra oil droplet worked for the favor of Alice Ball when she was working on it and was useful to the people who were suffering from leprosy. So now we know that the problem was not the biological activity of Chol Mogra oil. The problem was bioavailability, the ability of the drug to travel through the body. So Ball's task was now to make a hydrophobic drug behave in a hydrophilic environment without destroying the therapeutic part of the molecules. Chol Mogra oil isn't just one molecule. It's a mixture of molecules. And the two key therapeutic compounds are hydrocarpic acid and cholmogric acid. Both are cyclopentyl fatty acids, which has an unusual structure, but that structure interferes with the layer of Mycobacterium leprae. Now we need to understand the step 2, which is breaking the oil into usable components, which will happen via hydrolysis. We've already discussed how oil needs to be broken into substances which can interact with water easily so that it can penetrate the bloodstream or it can reach the bloodstream. Chol Mogra oil, as we know, it arrives as triglycerides. So triglycerides, what are these? I've attached a picture. It is 3 fatty acids attached to a glycerol backbone. To work with active medicinal parts, Alice first separated them through hydrolysis, which is breakdown of water. This step frees the fatty acid molecules, makes them available for chemical modification. All this happens without affecting their biological function. Now, the most crucial part was esterification to create ethyl esters. So this is the breakthrough. Ball converted these fatty acids into ethyl esters. Right, and how are ethyl esters formed? I've also attached a picture. So if you take a fatty acid, which is, let's say, an alkyl chain, R plus COOH, and along with you take ethanol, And then this forms ethyl ester, which is an alkyl chain, which is R, COO, CH2, CH3 plus H2O. So the new OCH2CH3 link is the highlight. So Ball converted fatty acids into ethyl esters, and it changed its mobility and its travel behavior. However, this then leads us to think, why ethyl esters? Ethyl esters are less viscous, which is it's thinner and easier to inject. They're more miscible with the aqueous environments, mixes easily, and when they reach the tissues, the body naturally cleaves, which is breaks the ester bond back into active fatty acid, which means that the drug becomes water cooperative for transport and then returns back to its therapeutic form inside the body. Now, the last step was formulating an injectable solution. As we already discussed, raw chol mogra oil is thick like cold honey. So the Ball method converts it into a liquid closer to oil mixed with saline, smooth, consistent, and safe to inject. The difference is the difference between viscosities, as in the biochemistry that happens inside the body. A quick recap as to what all the steps were. The fatty ester travels through the bloodstream. It then reaches the tissue, or the enzyme inside the tissue cleaves the ethyl ester, which restores the fatty acids, and it binds to mycobacterial cell wall, and it lyses the mycobacterial cell wall, breaks it down, and degrades the mycobacteria leprae.
So far, we have discussed all about what leprosy is, what are the clinical manifestations of the same, and all about the biochemical and biological aspects of the first treatment that was developed for leprosy, and the whole pathway as to how it functions inside our body. To conclude the video, I would love to add an animated YouTube video that I saw about the life story of Alice Ball and how she was a victim of Matilda effect, which is a term given to the process where the work of female researchers or female workers are discredited and the credit is stolen by other male counterparts. So this video will be the perfect concluding part or segment to our video for today.
Alice Augusta Ball was born on July 24, 1892 in Seattle, Washington to James Presley and Laura Louise Ball. The couple had 4 children. Her grandfather Ball Sr. was a famous photographer and one of the first African Americans to learn the art of daguerreotype. Her father, mother and aunt were also photographers. Alice Ball attended Seattle High School and graduated with honors in 1910 and went on to study at the University of Washington and earned 2 degrees, pharmaceutical chemistry and pharmacy. She transferred to the College of Hawaii, currently the University of Hawaii. In 1915, she became the first woman and the first African American to graduate with a Master of Science degree in chemistry. After graduating, she was offered a teaching and research position and became the first woman chemistry instructor at the age of 23. Alice Ball's research focused on finding treatment for leprosy. She worked extensively. She studied chol mogra oil and its chemical properties. Until that time, the oil was used as a topical medication and was not so effective in treating leprosy. Alice Ball created a water-soluble injectable form of the oil's active compounds that could be safely injected with minimal side effects. In Hawaii, those suffering from the disease were exiled to a facility called Kalapapa on the island of Molokai in leper colonies and kept away from families and friends until they died. Symptoms of the disease included scaly skin, lesions, with advanced stages producing limb deformities. Alice Ball's scientific research was so successful in treating leprosy symptoms for thousands of patients that it became known as the Ball method. Doctors used the method for years. It was so effective that leprosy patients kept isolated in medical facilities could return to their families no longer suffering from their symptoms. In 1916, Alice Ball died tragically at the age of 24 after accidentally inhaling chlorine gas while teaching in the lab. She never realized the full effect of her discovery. After her death, Dr. Arthur Dean, the president of the College of Hawaii, claimed her inventions as his own. However, in 1922, Dr. Harry T. Holman, an assistant surgeon at Kalihi Hospital, gave Alice Ball the credit she deserved in a published paper. Alice Ball's achievements have received more recognition. In 2000, former Lieutenant Governor of Hawaii, Mazie Hirono, declared February 29 as Alice Ball Day to honor Alice Ball's life and science achievements. In 2007, the University of Hawaii posthumously awarded her with the Regents Medal of Distinction. The Alice Ball Endowed Scholarship to support students pursuing degrees in chemistry, biology, or microbiology was established by scholar Paul Wermicher in 2017. Alice Ball overcame the racial and gender barriers of her time.
This brings us to the end of this video. I hope everyone liked and understood the video very well. I would like to thank the audience for being patient, and the organizing committee of Symposium IISc Bangalore, along with the respected judge panel who will be viewing this whole 15-minute video. Hoping that everyone liked it. Thank you. I would also be linking all the references at the end.